All right, class, last video for DNA this week. I'm going to show you something called the genetic code. Okay, so in the previous video, I like kind of piecing some of this together. So in the previous video, I showed you the idea of if we have this sequence of DNA here and we transcribe that into RNA, right? So DNA, mRNA. I was showing you that these bases in our mRNA get read 3 at a time, that those were our words in our messenger RNA. All right, so these words are called codons. Okay, so codons are pieces of messenger RNA, right? There are 3 bases in our messenger RNA. Why those codons are so important is because one individual codon is going to represent one specific amino acid. Right? 20 different amino acids. So one codon. So as we look at our mRNA up here, UAC. Remember, that's going to have instructions on what's the first amino acid. GUA, instructions, what's the second amino acid. All right, so we have these codons to make sure that we know exactly what amino acid we should be using. If we don't build the amino acid chain correctly, we can get a really screwed up protein that either functions poorly or doesn't function at all. So we have to be able to build the correct amino acid sequence. All right, so to do that, we use something called the genetic code. All right, this is a picture from your textbook. This is the genetic code. So what the genetic code does here is it's matching up codon to amino acid. Hey, there are 4 exceptions to this. All right, so sort of 3, technically 4. The 4 exceptions, and I'm gonna erase after I show you these because we need to be able to see this. But the 4 exceptions are this. So it's in green. This is what's referred to as a start codon. Okay, so it's actually always going to be at the beginning of our sequence. But it also does stand for an amino acid. MET is our amino acid. Okay, so there's one start codon, and then these 3 things in red up here, those are stop codons. Right, so like we can't just continually put in amino acids. We need to know where do we start, where do we stop. So there's codons that go for that. Right, in this genetic code here, let me show you real quick. So like these 3 letters here, I'm at the bottom right, those are our codons. And then this, in this case GLY, that's the abbreviation for the amino acid. Okay, so that's how this is laid out. Here's the codon, which is always going to be C's and G's and A's and U's, and then the abbreviation for the amino acid. All right, so that's kind of how this is laid out and what we're using it for. All right, so I do not have a sequence ready, so I'm actually going to pause this for a second. And I was going to say I'll be right back, but I hit pause before I said that, so I'll be right back. Okay, so sorry about that, but not like you really waited for anything. I learned the hard way that I have to pay attention to my sequences or I'm going to make something that doesn't work. So I'm going to show you one example of this, and then you're going to do one of these as the assignment part to this video. Okay, so here's our DNA sequence. we're just going to do a short piece here. So there's our DNA. So from there, we want to be able to come up with our mRNA, right? Basic complementary base pairs. Right, so there's our mRNA. So then now what we use the genetic code for, none of this is new, we've done this already, but what we use the genetic code for now is we look up these codons, these are now codons, and we try to figure out what is our amino acid sequence going to be. So only one time on this entire chart will we find AUG. They have this organized where there's first letter, second letter, third letter in your codon, right? So you can use the way they have it laid out like that, but like I just said, AUG is only going to be in this table one time, so you can really just look for AUG. All right, so we look for AUG, here's AUG, and then we want to write in what's our amino acid, MET, M-E-T. All right, so now we're building our amino acid chain. Next is ACG. We find ACG, there it is. We write in our amino acid, THR. We find CAU, here's CAU. We write in our amino acid, HIS. And then we get to UGA, which is here. And UGA is a stop codon. There's no amino acid there. So we would just simply write the word stop there. Right, so now we've done all of central dogma. We started with DNA, we made mRNA, and we made a protein. We made our chain of amino acids. Okay, so in the assignment for this video that goes along with this video, you're going to have to use this genetic code. Okay, so remember, when you see that, what you're going to see is a DNA piece. So you're going to have all this DNA. Remember then the first thing you're going to have to do is take that and make messenger RNA. And then you use that to come up with your amino acids, right? Use the genetic code to figure out your amino acids. And then I want to show you one thing real quick. You know, I've taught this a long time. So if I give you this DNA, notice our DNA here. I only gave you one strand of our DNA. That's all we need. So it's very common for people then, if I give you this strand of DNA, the first thing they'll do is write in the next strand of DNA, and then they'll use that to make the mRNA, and then of course they use that to find their amino acids, and all of that will be wrong. Right? So you don't have to do anything with the DNA. You don't have to build more DNA. You have your DNA, one strand, use that to find your mRNA, complementary base pairs, and then use the genetic code to look up what your amino acids should be. All right, so that's our central dogma, how we're doing genetic code, using the genetic code to build these proteins.